you are back, you can come back to secretion. There are secretory glands throughout the gastrointestinal tube. Whether they form multicellular glands or unicellular glands, they are all over the gastrointestinal tube. So there are a couple of things happening there, and I mentioned them already, but let's repeat them. So you have fluid, which on its own is important because particles are wet will the wetness allows some lubrication, so it flows better. But two, it will also have a certain pH, and digestive enzymes or other substances that are either important for digestion or for absorption. So let's go over all these levels of the gastrointestinal tube and discuss how secretion is controlled. So the salivary glands are located in the mouth. So let's review a little bit of anatomy. So you have the parotid gland, the submandibular, and the sublingual gland. Those are the major salivatory glands or salivary glands, but they are minor as well. There's a lot more than I could speak about the secretion, but we normally considered two types of secretion in the salivary glands. The alpha amylase, which is the ptyalin, the P is silent, and that's amylase is an enzyme that will digest carbs, the starches. Remember, this may be a good class for you to remember. The exam is cumulative. It's for you to go over the biochemistry component. So know what polymers are, polysaccharides are, and the monosaccharides. So as far as the enzymes, that's it. There will be more enzymes, but those are not relevant for digestion, meaning that they won't be able to contribute a lot through digestion. This is a serous secretion. Serous secretions, you may remember from your anatomy, they are more watery. But there's also mucus secretions, which are more sugary, and they're more helpful as far as lubrication. So let me just show you a little diagram of what a salivary gland looks like. So the primary secretions are ptyalin or amylase, alpha amylase, and mucus. And I mentioned the pH a couple of times. Let me just reiterate the pH here, but also add that the pH of the salivary secretions are around 6 and 7. So it means that the enzyme, in this case the alpha amylase, will act the best, will have higher activity at 6 and 7, between 6 and 7 pH, which another way of saying is that the optimal pH for the salivary gland, salivary enzymes, the alpha amylase, to act is between 6 and 7. So pause here if you want to just go back and forth, but we will proceed into the stomach. So the stomach has a series of glands. So it's like a sheet of cells that are all producing different components. We normally distinguish two types of glands: the oxyntic glands, also called gastric glands, and the pyloric glands. The pyloric glands are closer to the pylorus, so in the antrum, and they secrete essentially mucus and also a substance it's called gastrin. We'll see in a little bit what gastrin is for, but it will act essentially as a hormone and will help control different aspects of the gastrointestinal processes. The oxyntic glands are the ones that form acid. So they secrete the acid, which is the hydrochloric acid, and secrete an inactivated form of the enzyme pepsin, which is called pepsinogen. See, we've spoken about fibrinogen and fibrin, or plasminogen and plasmin. We also, in the very early lectures, we talk about a zymogen. So pepsinogen is the inactivated form of the protein digesting enzyme. The reason the cells cannot have activated enzyme inside of themselves is that inside of a cell we find a lot of proteins. So this would be suicidal for the cells. Okay. In addition, we mentioned the intrinsic factor, and of course they also produce mucus. All the glands in the stomach produce mucus. The oxyntic glands are found in the body and the fundus. I'm trying to go as slow as possible, but again, I will leave the pauses up to you. So this, you can even pause a little bit in a couple of slides, because the next slide we are expanding on the oxyntic or gastric glands. So the oxyntic and gastric glands have different types of cells. You have to know the last two. So we have mucus neck cells that produce mucus, but I want you to know that there are chief or peptic cells and parietal or oxyntic cells. The chief or peptic cells secrete pepsinogen. The parietal or oxyntic cells secrete hydrochloric acid and intrinsic factor. So now it could be a good time for you to see if you can know the distinction between the two glands, and within the oxyntic gland know the distinction between peptic and parietal cells. You have to know both names for each of the glands and also both names for each of the cells. The most important aspect of the parietal cell that I want you to focus on is the production of acid. And acid is produced via a pump called the potassium hydrogen pump, or hydrogen potassium ATPase, which is normally called the proton pump. You might have heard of omeprazole, and that's the PPI, a proton pump inhibitor. So it inhibits the formation of acid. So let's look at a picture of a parietal cell, and you see that we have the pump on the lumen side, lumen the inside of a tube, so inside of the stomach, and you see that it's sending protons and also sending chloride. These are the ingredients for hydrochloric acid. Note that for every molecule of hydrochloric acid we are sending into the lumen of the stomach, on the other end, on the basal aspect of the cell, we're actually sending one molecule of bicarbonate into the blood. This is important, but we are not going to fully explore all these components. It's important to understand acidosis and alkalosis. For example, with if a person keeps vomiting, they will go into alkalosis. They they are vomiting out acid content, and they will produce more acid. And remember that you're also producing bicarbonate. So more bicarbonate is a situation of of alkalosis. Bicarbonate is metabolic, so it will be a metabolic alkalosis. So the pump is an element that is very important to understand, because if you think about gastroesophageal reflux, you think about ulcers, a great part of it has to do with the damage that is caused by the acid. Normally the stomach is somewhat protected against the acid by a series of components. Not only produces bicarbonate underneath, which is alkaline and may neutralize the acid, but it may also has tight junctions, profound perfusion. So all those aspects are very important for helping out the stomach resist any acid. But in many diseases, the mainstay of treatment is controlling the acid, and that's why it's important to know the proton pump inhibitor. And to further understand this, we need to look at the aspects that are stimulatory or stimulating or inhibitory as far as the parietal cell. So remember that antrum produces gastrin. You see here how gastrin stimulates the parietal cell to produce more acid. So this is the kind of hormonal control that happens. So the antrum is telling, quote unquote, the upper portion, the parietal cells, to produce more acid. Histamine also helps out and stimulates the cell, the parietal cell, to produce acid. Different receptors than the receptors that we use for example, allergic reactions, but they are important. And remember, the parasympathetic is rest and digest. The parasympathetic uses acetylcholine, and you see acetylcholine as a positive effect on the gastric cell. So I would want you to understand those are all positive aspects, but you see the somatostatin, the same thing that inhibits the growth hormone, the somatostatin acts in many aspects of the body, including here to inhibit the parietal cell. Okay? I wanted to cover a little bit more about the parietal cell, and then we go to the chief cells. And what I wanted to cover is the intrinsic factor. So I would like you to relate this with the vitamin B12. So the more you connect these dots, the easier it is for you to go over all the material when it comes to a cumulative exam. Depending on how prepared you are, it may not be, it's a little bit, unheard of for you, but perhaps you could pause here and even go through the slides of anemia and understand that vitamin B12 absorption depends on intrinsic factor. And the slide shows you something called pernicious anemia, which is a situation in which our parietal cells will not, or the intrinsic factor will not be able to perform its function. So a lot of the vitamin B12 is going to be is not going to be absorbed. So let's just one more slide about now about the chief cells to speak about pepsinogen. Pepsinogen, we said everything that is written on this slide already, but let's just say it again. Pepsinogen is an inactivated form. It has no digestive activity. It needs to be activated into pepsin, which happens because of the acid. The pH in the stomach is acidic. We talk about something below 3.5. This is enough to neutralize the amylase. Remember, the amylase is a protein, so it will be digested, but also the amylase has a different pH, which is slightly more, which is not basic, but is slightly acidic, whereas a pepsinogen is very highly acidic. And so you could stop here and just review all the anatomy of the stomach. I wanted to just tell you a little bit, introduce the topic of how the gastric secretion is controlled even before we eat. So we are used to, if you think about food, if you go into a restaurant, if you look at the kitchen food, you may actually notice that you produce more saliva. But in fact, we also produce other secretions, such as gastric secretion. This is called the cephalic phase of gastric secretion, so the head phase of gastric secretion. Then when the food is actually in the stomach, it activates the stomach to produce more. But even after the food exits the stomach, so at that point we call the chyme, there's some secretion. So the diagram on the right is just to help you understand that, but you can connect that with the type of receptor. So we have gastrin, which is stimulatory, histamine, which excites the parietal cell, but somatostatin is an inhibitory component. So I would pause definitely here, because this might have been a little bit. It's a lot of information for you to go without pausing. So I would pause here, but if you want to proceed, or when you come back. Proceeding now to the pancreas. The pancreas has both exocrine and endocrine functions. We'll speak later on about the endocrine functions, but the exocrine, which represents that it sends its secretions into a place other than the blood, is responsible for several aspects of digestion. So up to here we spoke about the mouth, we spoke about the stomach, and now everything else that involves digestion will happen in the small intestine, and will be in great part with the help of the pancreatic juice. In addition to having enzymes, it will have a lot of bicarbonate ions. Bicarbonate will be responsible for neutralizing the acid. So all this, the enzymes and the sodium bicarbonate, will flow through the pancreatic duct, which then will join with the common bile duct. And all together they will penetrate the duodenum in a structure that is sometimes called the ampulla of Vater or papilla of Vater. And it has a sphincter, which is called the sphincter of Oddi or sphincter of the ampulla. So let's start with the enzymes. So virtually the pancreas provides also all the enzymes to digest everything, from carbohydrates, lipids, and proteins, but also aspects such as DNA and RNA and so on and so forth. Let's focus on the most important ones. So for protein, the pancreas will produce enzymes that are also inactivated, but the final enzymes are trypsin, chymotrypsin, and carboxy polypeptidase. The most important one is trypsin. So this will take care of the proteins. So the stomach already start digesting proteins, and so the final digestion will happen now at the level of small intestine, in which proteins will be further broken down into peptides, and the peptides will be further broken down into small peptides and amino acids. For carbohydrates, the pancreas has the pancreatic amylase, which would have the same function in relation to the starches. So this is a little bit of a dynamic process. It doesn't mean that the digestion is 100% efficient in the sense that every protein is broken down into amino acids. So it will be very dynamic. Some protein, depending how fast the digestion is, for example, some protein, some carbs, some lipids may go undigested or may be digested only partially. But we are talking about the general idea. So the carbs will be digested into monosaccharides, and that happens through the amylase. But then we'll explore that it will require further enzymes, because normally the amylase breaks down into disaccharides. For lipids, we have the lipase and other ases, but that would be for cholesterol, for example, cholesterol esterase, phospholipase. So the triglycerides are transformed into fatty acids and glycerol. So it's again a good example for a good timing for you to review lecture 1 as far as the molecular biology, as far as the nutrients. So let me just talk about the pancreatic enzymatic activation. So again, the protein digesting enzymes are produced in an inactive form. So we'll have trypsinogen. Trypsinogen is the first enzyme to be activated. It is activated by enzymes on the luminal membrane of the cells, what we call brush border. Remember, I asked you to focus on to look at the label that said brush border. Go back if you need. And so once the trypsin is formed, it will activate all the other protein digesting enzymes. So we have here a system that not only has secretion, but also all that secretion has fluid and has enzymes. You saw a lot of pumps working, so this process is expensive. So we surely want to have this very tightly controlled. And so the control is done by local factors and systemic factors. So the parasympathetic has a general activation over the digestive system. And we have acetylcholine activating the pancreatic secretions. But then we have two other substances which act as hormones, because they will produce release in the blood, and then will act in different portions of both the pancreas, the gallbladder, and the sphincter of Oddi. So let's put this together, and again, there are several things we could say about these two components, but we are going to be very specific, so pay very close attention. The first component is called cholecystokinin, CCK for short. CCK is secreted by the duodenal and upper jejunal mucosa. When? So you have to know the local, the place, and then what is the trigger and what is the function. So the trigger is food. We'll see what kind of food, but it's essentially fats and amino acids that cause the release of CCK. Secretin is another hormone that is also produced by the duodenum and jejunum, but in this case is when acidic food enters the small intestine. And so you interpret this like you interpret hormones. So you have a problem that triggers the hormone, and then the hormone should help solving the problem. So the problem of food arriving into or entering the small intestine is that we will need enzymes, and we will need other substances that are required to help digest food. The problem, quote unquote, of acidic food entering the small intestine is that we have the pH is too acidic, so we need to neutralize. So the CCK acts on the pancreas essentially to stimulate the secretion of enzymes. And will act also on the gallbladder and on the sphincter of Oddi so that it relaxes more of the secretion that is stored in the gallbladder, that's called the bile. Secretin, on the other hand, has an overall effect of stimulating the secretion of the pancreas, including bicarbonate, which should neutralize the acid and therefore create the regular pH for the pancreatic enzymes to act. I have more, we'll come back to this, but pay close attention to this, pause as you see fit. We surely will come back to this topic. So let's talk about the liver now, and that way we'll complete the picture. Is the last one, the last organ that contributes to digestion. Not only we've talked about the liver, not from different points of view. We talked about the liver as far as metabolism, so when it comes to gluconeogenesis, glycogenolysis, and so on and so forth, so all the metabolic aspects of the body. We spoke about the liver when we talk about producing proteins, such as albumin, such as clotting factors. Now we need to talk about the liver as a secretory organ. And we need to talk about the bile. So the bile plays a very important aspect in fat digestion and absorption, but is also a means of excretion. So the bile is produced by the hepatocytes and will contain substances that the body wants to excrete and contain substances that will help with digestion. The substances that help with digestion, for now, we can call bile acids. We will talk a little bit more about this, and we'll talk also about lecithin. But essentially, the liver, the bile acids and the lecithin are responsible for emulsification. Let this word sit here because we'll come back to it. But emulsification, for now, just imagine what happens when you put dishwasher into the dishes. So it breaks down the fat into small particles. This will be important for digestion, but also for absorption. The bile, as far as excretion products, will focus essentially on bilirubin. So let's work on bilirubin, and then we'll come back to the bile acids and lecithin when we talk about the fat. So bilirubin comes from the degradation of hemoglobin or any other heme proteins. So we degrade bilirubin, hemoglobin, because the red blood cells die, or because the red blood cells at the factory, there's something wrong about the hematopoiesis, that a lot of red blood cells are being destroyed, or they're precursors. In any case, the heme has iron, and we talked about it in the fact that we need to preserve that iron. But what to do with the rest? The rest becomes biliverdin. Biliverdin is transformed into bilirubin. This bilirubin is not soluble in water, and we call this bilirubin unconjugated. This bilirubin needs to travel the blood bound, or most of it travels bound to proteins. The most important, albumin. So once this albumin and bilirubin are in circulation, the hepatocytes will uptake it, will take it in, and will transform this unconjugated bilirubin into conjugated bilirubin, which you don't have to know the enzymes, but you see there number 4, the process of conjugation. Conjugated bilirubin is now water soluble and will be excreted into the bile, and it's stored in the gallbladder along with the other bile, and then it will be excreted when the time comes, secreted or drained into the small intestine. Note that once in the intestine, a certain amount of this bilirubin, it will be converted into something called urobilinogen. You notice that uro stands for urine. Sometimes, particularly in the old books, old textbooks, people like to talk about sterco. Sterco stands for feces. I have a picture that will show that. But the urobilinogen or stercobilinogen are the same thing. So part of this urobilinogen may actually be reabsorbed. How is it reabsorbed? Well, there are enzymes that will hydrolyze Note that not all bilirubin that is excreted into the intestine will be sent out into the feces. Some actually comes back to the blood. This is what we call the enterointestinal hepatic circulation. And some of this urobilinogen that is reabsorbed actually may be showing up in the urine. That's why it gets the name urobilinogen. Urobilinogen. Okay, so basically this is a very important cycle. You can use this picture, or to pause here and use this picture and see if you understood all the details, or use, for example, this picture. So heme, biliverdin, unconjugated bilirubin, conjugated bilirubin, urobilinogen. Urobilinogen, once it's oxidized, becomes urobilin. That's what gives color to the feces. Some books like to call this stercobilin, and particularly if they call stercobilinogen, and that sterco stands for feces, and urobilin stands for urine. That gives the brown color both to the feces and the urine, but that's a detail. Okay, good time to pause here. So what is jaundice? Since we are on the subject of bilirubin, jaundice is the yellowish color that is noticeable, best noticed in wide areas, such for example the sclera, or people who have a very fair tone of skin, and it's caused by the accumulation of bilirubin. This is just a review of the portal system, just to understand that the liver gets all the blood that comes out of the, virtually all of it. It comes out of the small intestine, this is just to remind you of the hepatic portal system. And understand that the liver gets the blood from the portal vein. The portal vein carries a lot of the blood from the gastrointestinal tract, but the one that we are mostly interested in is the blood from the small intestine, because this blood now has all the nutrients, and so the liver is in this perfect place to control the metabolism of the body, because now it senses what is in the blood that just came out of the intestinal tube, and therefore that just reflects the food that we ate. This is also a reminder of your anatomy. Just here we have the hepatocytes. It's a good reminder that the hepatocytes are the ones that produce the bile. So the bile flows into hepatic ducts. And then you may have something called a common hepatic duct that then merges with the cystic duct from the gallbladder, and altogether they form the common bile duct. There are some anatomical variations, but for us we would the most important or the more common one is the idea that the main pancreatic duct with the common bile duct drain together, and that there is a sphincter called the sphincter of Oddi. See the representation of the enterohepatic system, in which there is reabsorption of some of the bile products back into the intestine. So once things are found in the intestine, the intestinal cells don't know what was meant for excretion or not. There will be a simple absorption. So let me just go back now to the issue of CCK and understand how CCK acts on the gallbladder and the sphincter of Oddi. We could speak about many more factors or effects of CCK. Let's just keep this one. So the presence of food in the duodenum, particularly the fatty acids, will stimulate CCK. CCK does several things again, but just focus on these two. Contracts the gallbladder, so it's going to, by contracting the gallbladder, remember the gallbladder is smooth muscle, it contracts and may increase the amount of bile that is drained into the duodenum, and simultaneously opens up, dilates the sphincter of Oddi by relaxing the muscle. Simultaneously, this will allow more bile flow into the duodenum. So now it's a good time for you to pause and understand the role of CCK and secretin. Again, your goals would be, or are actually, to know what are the stimuli, where are these hormones produced, and what are the overall effects. To finish this part, we just need to talk about the intestine. The small intestine contributes to absorption. It's the place where digestion takes place, and we also have in the on the membrane of the enterocytes, we find further enzymes that help with the digestion. Then the large intestine may secrete mucus, and also likewise the small intestine as well, and may also reabsorb water and electrolytes. I wanted to just really put this slide here to help you understand that the enterocytes have a brush border and will have brush border enzymes such as lactase, for example, which will help finish the process of digestion. We'll see how it works now for all the nutrients in a different video.